In this video, we will appropriately describe when biochemical or cell-based assay formats are applicable for HTS. So when it comes to high-throughput screening, HTS, choosing between biochemical and cell-based assay formats really depends on the kind of information you want to get from your screen. Biochemical assays are great when you want to study the direct interaction between a compound and a purified target, like an enzyme or a receptor. These assays are typically simpler and more controlled because they use isolated proteins or molecules, so you can easily see if your compound has a direct effect on the target's activity. They're especially useful early in drug discovery when you want to quickly identify hits that modulate the function of a specific protein. On the other hand, cell-based assays are more complex and biologically relevant because they involve living cells. These are ideal when you need to see how compounds affect cellular pathways, processes, or phenotypes. Cell-based formats allow you to capture effects that depend on cell permeability, metabolism, or even toxicity, things you'd miss with a simple biochemical assay. They're particularly valuable if your target is part of a larger signaling network, or if you want to observe functional outcomes like changes in cell growth, gene expression, or morphology. In short, use biochemical assays for straightforward, target-centric questions and reach for cell-based assays when you need to understand how compounds behave in a more realistic biological context. See you in the next video.